So my lab currently is working on, so we sort of started, focused on type 1 versus type 2 CALR mutations. Now we've broadened more to all driver mutations and MPNs and trying to understand what are the dependencies specific to different cell types. So we're really interested in like metabolic dependencies of the mutant stem cell compartment, and then as the cells differentiate, how do their needs change? Like, how does metabolism change? How does immune surveillance change? And how can we use that information then to eradicate those cells, which in theory would be curative? So that's our goal, is to really like deeply understand the biology of these different cell types and use that information to be able to completely get rid of those cells from the body.